Okay, so this is cellular respiration, electron transport chain, lesson 5.5. All right, so let's just review a little bit what we have done so far. So glycolysis gave us 2 ATP. It also gave us 2 pyruvic acid molecules that went into the citric acid cycle. Those 2 pyruvic acid molecules were oxidized and used in a series of reactions to create more products. And in glycolysis, we also had 2 NADH molecules that went into the electron transport chain. The citric acid cycle also produced 8 NADH molecules and 2 FADH2 molecules. Remember that FADH2 molecules are really similar to NADH in that they're carrying a pair of high energy electrons that can be used in the 3rd stage. So we're going to get a lot of ATP out of the electron transport chain. And do you guys remember where it happens? Yes, where in the mitochondria? Not the matrix. The citric acid cycle happens in the matrix. Yes, it happens right on the inner membrane, and it's a series of basically transports that are going to move things back and forth across that membrane. So we'll talk about that today. Okay, so electron transport refers to using high energy electrons from the citric acid cycle and glycolysis to convert ADP into ATP. So remember that when ATP is used for energy, it's just breaking off that last phosphate bond, and it turns the molecule into ADP, which is adenosine diphosphate. So if we want to convert that ADP back into ATP, what do we have to do? Like, what turns ADP back into ATP? Yes, adding an additional phosphate to the end of it. So that's what the electron transport chain does, is provides energy to put that last phosphate bond back onto ADP, so then we end up with more ATP. It occurs in the inner membrane of the mitochondria. Oxygen is required to be the final electron acceptor. Oops, sorry. So those electrons have to go somewhere, so the oxygen is going to accept it at the end. So that's why it's considered an aerobic reaction. good on this slide? Okay. So this is a picture of it. You guys should have this in your diagram, I think, in the packet that I gave you. But basically, the mitochondrial inner membrane contains numerous protein carrier complexes. Remember when we talked about the different types of cellular transport? We talked about how it's basically the fluid mosaic model. So mosaic referring to the fact that membranes have a bunch of different protein complexes in them. And in this case, these protein carrier complexes will be used for the electron transport chain. So it's just like the name sounds. So electrons are going to be transported in a chain throughout all of these different molecules, and energy will be transferred to be able to put ADP, or to turn ADP back into ATP. So this is where we're going to bring back some of those concepts from cell transport to talk about what is really happening here, whether it be facilitated diffusion, active transport, or just regular diffusion. Okay, so also NADH and FADH2 that was produced in the citric acid cycle are in the mitochondrial matrix. So that's in the middle, right? Because that happened in the matrix, so that's on the inside of the inner membrane. So remember that. Oops, sorry. Look at the last page of your packet of the diagrams that I gave you. It is this, right? Yeah, okay, good. So this is the very outer membrane of the mitochondria, and this is the inner membrane where all of the proteins are. What? So that all of this can happen. So it has the outer membrane that encloses the mitochondria, and then the inner membrane where everything can happen. Okay, so high energy electrons from NADH and FADH2 are given to the protein carriers that are embedded in the inner membrane. NADH goes back to NAD+, and FADH2 goes back to FAD. So the difference between these two molecules, NADH and FADH2, is that they will go to different carrier proteins in the membrane. So that's why they're different, because they're going to go to a different place. Where they're going to give their electrons to a different protein in the membrane. So NADH and FADH2 are the molecules that have the high energy electrons, and when they give them away, they turn back into NAD+ and FAD. And remember that we created 2 NADH molecules in glycolysis. And then in the citric acid cycle, we created 8 NADH molecules and 2 FADH2 molecules. And that's per glucose. So the first thing that happens is that NADH will come up to this protein, FADH2 will come up to this protein. They're going to give away their high energy electrons, and then those high energy electrons are going to move through in a chain, and I'll tell you how that's happening. Yes. Uh-huh.
The mitochondria was like absorbed by some prehistoric cell, and they like started working together to create the animal cell.
Yes.
Does the mitochondria have the this entire process, like the tubes, the membrane, and the electron transport, because it did it itself in the cell that absorbed it?
Yes, it could have. The mitochondria prehistorically could have existed on its own and was able to do this process on its own without being inside of a cell. Engulfed it and became more complex. The endosymbiotic theory. Okay, yes. Wait, say that again?
Because you said that the mitochondria is engulfed by the cell and more complex.
Uh-huh. Or mitochondria what? What is the last part? Engulfed? No, they are now made inside the cells. Like they became a part of the DNA of those cells that took them in. Okay, so next thing that happens, those electrons from NADH and FADH2 are passed along the electron transport chain from one carrier protein to the next. As they are passed along, the electrons go from a high energy state to a low energy state. Oops, sorry. So as they move through this, they lose their energy basically, and the energy is going to be used somewhere else to do something else. I'll tell you what that is. So these are the different protein carriers, obviously, and the electrons move through them. And as they do, they lose their energy. They're passing their energy so that other things can be done. You don't need to know the names of all of these proteins. You just need to know that electrons are passed through them. And in a little bit, you'll understand what that energy is going to be used for. Okay, we good? Okay. At the end of the chain, an enzyme combines with these electrons, or combines these electrons with hydrogen ions and oxygen to form water. What's happening right here? Oh, whoops, sorry. I've gotten questions on this before of like, why do we need to drink water then if we can make water during this? It doesn't make near enough water that you actually need in order to survive. So it's just more of a byproduct. Okay. Yeah. Yeah, because if you're in a coma and you're in a hospital where they're still taking care of you, they're giving you still the nutrients and the things that you need through an IV or a feeding tube. And then if you need to be on a ventilator, that's what's supplying your oxygen. So yeah, it's still happening. It's probably not happening as fast because you're not using as much energy, but it still is happening. Okay, so as high energy electrons move to a lower energy state, their energy is used to move hydrogen ions across the membrane. So hydrogen ions are going to move from the mitochondrial matrix into the inner membrane space, okay? So this is going to be an example of active transport because there's going to be a high concentration of hydrogen in the inner membrane space and a low concentration down here. So if we're moving from a low to a high concentration, that's going to require energy, right? So this is an example of active transport. So the hydrogen ions don't naturally want to be up here. They want to stay down here because they want to try to reach equilibrium, right? But they're going to be pushed to a higher concentration for a specific reason, which I'll tell you about in a second. So hydrogen has a couple different roles in this process. Hydrogen is going to combine with oxygen and the high energy electrons to produce H2O to be that final electron acceptor. But then it's also going to be used to do something very important, the whole reason why we're doing this, which I think is on the next slide. You good? Oh, sorry. One last thing. I forgot that was on it. Active transport, because we're going from low hydrogen to a high hydrogen concentration. Okay. So the inner membranes of the mitochondria contain protein spheres called ATP synthase. So there's this protein that's at the end of the electron transport chain. This is the one that you do need to know what it is and what it's called. These ones you don't. These ones do have their own names and are specific, but we don't need to go into that much detail. You do need to know the ATP synthase molecule though. It's a membrane protein and it's going to do a very important job. So all of this hydrogen that was pushed into the inner membrane space where there's a high concentration, what do you think it's going to want to do? Yeah. What? Yes, it's going to want to go back. So I'll give you a couple more seconds on this slide to write this down. So the whole reason why it's pushed into the inner membrane space where it doesn't want to be is because naturally it's going to want to go back to the lower concentration by diffusion, right? So the only place that it can get back through to where it wants to be is through the ATP synthase. And when that happens, actually write this down too. Sorry, I think my words are getting ahead of the text that's on the screen. So the inner membrane is impermeable to hydrogen ions, but they want to diffuse back across into the matrix. The hydrogen ions diffuse through channels in the ATP synthase. Actually, I'm on point. Okay, so the hydrogen was pushed into the inner membrane space from a high to a low concentration. So it doesn't want to be here. It wants to go back through this membrane. But the only way back through is going to be through this ATP synthase. And each time it goes through the ATP synthase, it's going to spin it, and that spinning of the ATP synthase is going to provide kinetic energy to add phosphate back to ADP to make it ATP. So I'll tell you how that happens on the next slide too. Okay, we good here? Okay, so the diffusion of hydrogen ions through channels in the ATP synthase cause the ATP synthase to spin. So this is going to be passive transport though, because it's moving from high to low concentrations. It is facilitated diffusion though, since it's moving through a transport protein in the membrane. So a test question that you might have is the movement of hydrogen ions through the ATP synthase can be best described as, the answer would be facilitated diffusion. Okay, we good? Okay. So as it rotates, the enzyme, ATP synthase, uses that kinetic energy to attach a phosphate to a low energy ADP, forming high energy ATP. Oops, sorry. So ATP synthase is special because it's an enzyme. As you can tell, it ends in the letters ASE. So it's an enzyme. It's carrying out a reaction, but it's also kind of considered a carrier protein because it's embedded in the membrane and it's going to provide a channel for the hydrogen to get through by facilitated diffusion. Question? A phosphate. I don't know actually. Is your diagram of this blank? Like, are there spots to fill out? It's on the very back page. Are there blanks? It's all filled out for you? Okay. I'll give that to you tomorrow. Okay, so just another picture of it. Another picture of it. So we can fill this out now. So it happens in the inner membrane of the mitochondria. It is aerobic. It doesn't have a starting carbon-based molecule or an ending carbon-based molecule, but it produces 32 ATP. So obviously that's the bulk of where ATP comes from. So if for some reason we can't do the electron transport chain, then we're missing out on a lot of ATP. The electron carriers that it produced, instead of producing the NADH and FADH2, we took away high energy electrons from these molecules. So instead we produced 10 NAD plus molecules and 2 FAD. And then other molecules that are produced are H2O as the final electron acceptor. Hmm? Yes. Yes. Yeah, it's cloudy outside. Okay. That is it. Yes. Huh? Yes, you can work on homework. I'm gonna see if there's a video I can show you guys on this. I'm pretty sure there is.